to talk about vaccines, monoclonals, and this virus that's caused the pandemic. Now, in order to address that, I want to start with a little introduction about pandemics. And I would like to go back historically to 2009. In 2009, we had another pandemic. It was caused by avian influenza. Fortunately, was not as bad as this one, but it was the first time that we really tried to make a vaccine, and in a very short period of time in order to respond to a pandemic. And what happened was that day zero, basically the virus, the avian influenza virus H1N1 was isolated, and immediately started the race to make vaccines. Now, in 2009, making vaccines meant that the virus was isolated, was sent to Center of Disease Control in United States, or NIPS in the United Kingdom. And there, the virus, the avian virus, was adapted to grow in eggs. And then this took a couple of 3 months. After it was adapted to grow in eggs, basically the virus was sent to the vaccine manufacturers, and we and many other vaccine manufacturers started to make a vaccine. And we were very proud because in 6 months later, the first doses of vaccines were made, and basically shortly after, we got a lot of doses. But unfortunately, the virus was faster than we were. So basically, you see this gray line basically is the peak of the virus. The virus peaked much earlier before we had the vaccines. So we are proud to make vaccines, but the vaccines had no impact. Fortunately, the pandemic was not that severe, so we survived that. But the question was, the message was clear. I mean, our way of making vaccines was not fast enough in case of pandemic. So that was in mind. In 2011, I published a paper in Nature and addressing these things. And this is what a figure from that paper. Basically, I was trying to see how long it was going to take to make vaccines. And I looked back in 1980. 1980, basically, to make a vaccine, we did not have many technologies. To discover a vaccine took a very long time. Development at that time was easy. Well, it was easier. With 600 people, you could get a license. And so basically, it was pretty fast and relatively inexpensive. By 2010, a lot of things had happened. A lot of technologies, mostly genomics, all the omics, the ability, new adjuvants, basically that accelerated enormously the discovery. Now we could do not one antigen at a time, but with using the genomics and the omics, we could do many, many things in parallel. So basically, instead of taking a long time, discovery was much faster. But in the meantime, development had become much longer and much more expensive. The reason for that, well, many things, including the introduction of good manufacturing practices, good clinical practices, more requirement for safety. So basically, was much longer. So at that time, in 2010, I made the prediction that the same technology that had been so important to shorten the discovery by doing things in parallel, the science was going to also affect the development. So we'll be able, and I said in 2020, basically, vaccine development will be shorter because we are going to do a lot of things in parallel because the science is going to help us. So this was the prediction. And obviously, I did guess right that the 2020 things were going to be shorter. But my prediction was going to be basically shorter, but still taking years. Indeed, in 2020, when the pandemic came, it took less than a year. So what has happened in the meantime? Well, in the meantime, basically, really the science, there were a lot of advancements in science. And basically, all the systems biology, the molecular biology, the ability to do biobanks, have biomarkers, new technologies for manufacturing, and complete revolution in doing clinical trials. Basically, could be applied to basically shorten the development timelines. So in 2019, we had the meeting, and we said that the technologies were there, should be able to make things shorter. But in fact, by 2019, we're still doing vaccines in a very long time. And because there was no rush, no need to, we had the technology, but not really need to shorten. But then the pandemic arrived, and we did very, very fast. And the question is, obviously, why were we able to go from 10, 15 years to less than 1 year? It's an incredible achievement. And the reason, well, two main reasons. I'll go through them. One is basically the technologies, obviously. And the other one is the investment from the public sector. So I'll talk first about the technologies. In this slide, you see an overall summary of all the technologies that have been used to make vaccines. Initially, vaccines were made very crude, empirical, with an empirical approach. Basically, what you're doing, take bacteria, viruses, parasites, you kill them and injected them, or you attenuated and injected them. Very crude vaccinology. Then 1980 started new waves of technology. First was recombinant DNA, glycoconjugation. Then we came with the genomic approaches. And today, if I look at the technology today, I see an explosion of new technologies. And so much that basically today we can do things we couldn't do 5 years ago. And probably in 5 years, we'll be able to do things we cannot even dream about today. So anyway, in this explosion of technologies, the 4 of them were essential to make vaccines for COVID-19. And these are, one is genomics, reverse vaccinology, that allowed what I call internet-based vaccines. I will have a separate slide about that. The second one was the structural vaccinology, the ability to basically get the molecular structure, X-ray structure of proteins and design the stability and the immunogenicity of the proteins by modifying the protein by structure-based design. The third one is the synthetic biology, mostly RNA vaccines. And the fourth one is the availability of adjuvants. So let's start with this one, the first one, which is genomics and internet-based vaccines. To explain that, I need to go back 2013. 2013 was the Easter Day. And Easter Day, the Chinese, the Center for Disease Control, put in the internet the sequence of a new influenza virus, avian influenza virus, H7N9, that already killed 3 people in China and was basically predicted to be a new pandemic strain. And so as soon as the sequence came, obviously, everybody rushed again to make a vaccine. And most of the people, actually all the people, started to try to get the virus from China to Central Disease Control in the United States and NIPS in the United Kingdom. It took a long time to get there and adapted to the conventional ways of making vaccines. And that was taking months. We did a completely different thing. So as soon as on the day, Easter Day, the sequence was already in the internet. And on Monday morning, Craig Venter in San Diego in California was collaborating with us, used the genomic sequence to synthesize 2 genes necessary to make a vaccine. Monday night, those 2 synthetic genes were shipped by express mail to our laboratories. They were in Boston at that time. Tuesday morning, those synthetic genes were used in our laboratories to make 2 types of vaccines. One was a fully synthetic RNA vaccine. And in 7 days, we were able to make that vaccine ready to go into animal studies. The other one was a more conventional vaccine. Basically, we used the synthetic genes to generate, by reverse genetics, a new influenza virus able to grow in eggs, and then we could make vaccine. So basically, in 1 week, we were able to make 2 types of vaccines, a new one based on RNA, one more conventional one based on a real virus. And we had done that without ever seeing the virus, without shipping the virus. And basically what had happened was that instead of shipping the virus across the world, we're shipping the information through the internet. Somehow the information teleported through the internet was being used, and we did not have the need to look at the virus. So that moment is what I call the transition between what I call analog vaccines to digital vaccines. What's the difference? Well, analog vaccines is what we used to do. You take a virus, you take a bacteria and a parasite, you need to grow in a fermenter, and then, or you take a recombinant organism, you need to grow in a fermenter, purify the things, and then you make a vaccine. You need the real material, you need the real stuff to make a vaccine. With digital vaccines, basically, what you get is someone is going to do the sequence of your pathogen somewhere in the world, is going to send that through the internet. The information comes to a computer. Through a computer, basically, you design the vaccine and you send the information to a robot that will make a synthetic gene. And in the case of RNA, you do inject a synthetic gene, which is actually not the vaccine. It's the code to make the vaccine. And used by our cells to make their own vaccine. So I think the analogy between analog vaccines and digital vaccines is pretty high. And that indeed what really it can do today with the technologies. An extension of that is that what's going to happen in the future. In the future, basically, you will have this sequence. You have sequences, pathogens will be sequenced, bacteria, virus, parasite, worldwide. The information about the sequence will be teleported through the internet. Vaccines will be designed, and the information, basically the computer can send the information of the vaccine to many robotic stations worldwide, and this robotic station will make a vaccine locally. You can even think that, let's say 30 years from now, when we're going to have a colony in Mars, at that time, if you want to put vaccine on Mars, basically you have 2 choices. You make the vaccine physically in our planet, and then you send that a shuttle to Mars. It's going to take 9 months to get there, or you send the information at the speed of light through, and then you have basically a robot in Mars in 2 days going to make a vaccine. So, I mean, this seems a little bit kind of science fiction, but the only science fiction for the moment is really the colony on Mars. All the rest is actually pretty real. So that's the technology that can make things very fast, and this idea of digital vaccines is really important. The other thing which I said is very important to make vaccines fast was the investment of the public sector. Why is that important? Well, in order to explain that, let me tell you how we make vaccines. To make vaccines, the first thing you need to do is discover. I mean, you need to discover something. Once you have a good candidate, then you basically start what we call early development, and you go scale up. You manufacture, you do phase 1 clinical trials, phase 2 clinical trials. Once things work there, you go into the late development. Late development is phase 3 clinical trials, building a manufacturing plant, doing all the safety, all the things that, and all this exercise takes more or less 1 billion of investment. Obviously, I mean, And this billion is spent 10% in discovery, 20% early development, 70% in the late development. Clearly, in companies, we never spend the 20% of early development unless discovery is successful. We never spend 70% of late development unless we are early development successful. And this is because obviously we need to mitigate the risk, because if we spend 1 billion upfront, we're going to go bankrupt pretty quickly. So that's the way we make vaccines, and obviously it takes a long time. We mitigate the risk by doing things sequentially. Well, this time we had a pandemic. So in 2020, when the pandemic started, the US government, BARDA, with the Operation Warp Speed, basically they put 13.5 billion on the table, and then Europe, UK, and Japan put other money for a total of 15 billion. With that money, the public sector, well, mostly the American government, came to companies and said, look, companies, I know you usually do development this way: discovery, then phase 1, then phase 2, then phase 3, and you take 10, 20 years. I said, and they said, we cannot wait there. I know you do this because you need to save money to mitigate the financial risk. But they said, this time, I give you the money. So the public sector, mostly the American government, gave 1 or 2 billion per company. And they said, this is the money. You are not making any risk. You don't risk any money. But now you do development in a totally different way. So what you do is that you start discovery. I know discovery is going to last a few months, maybe a year. But as soon as you have the first candidate very early, you move to phase 1. And I know phase 1 usually lasts 18 months. Don't worry. As soon as you have the first thing that the vaccine is safe and immunogenic, you move to phase 3. And then phase 3 is going to last 2 years. Forget it. As soon as you have first data, the phase 2, you move to phase 3. And basically, by doing that, we were able to make vaccines in 10 months instead of many years. And the secret was doing things in parallel. Now, we did things in parallel because the public sector took the financial risk. Companies were not going to be able to take that kind of financial risk. But at the same time, it's also clear that we went fast without taking any risk on safety and efficacy, because all the steps were done. They were done in parallel instead of doing sequentially. But we did all the steps to establish safety and efficacy of the vaccines. So that's why we went so fast. So what happened? Well, what happened was, the computer doesn't like to move to the next slide now. Let me see. No, I guess I need
getting
to
out of the presentation mode and start it again.
Yeah, I'm trying to exit from the presentation mode, but doesn't want to do it. My computer got stuck. I'm sorry about that. Okay. Okay, I'm going to go back to the presentation mode now. When I get a pointer, okay, hopefully, okay. So what happened? Basically, what happened was January 10, 2020, the Chinese Center of Disease Control put on the internet the sequence of the new virus, SARS CoV-2 virus. Immediately, this time there were more than 300 labs worldwide that downloaded the sequence and were able to make synthetic genes. And then the synthetic genes were mostly the synthetic gene of the spike protein of the virus. Then they were used to do several types of vaccines. One was using the synthetic genes to make messenger RNA, and basically the messenger RNA is all synthetic. You take the template of the synthetic gene, you basically make the in vitro transcription, and you have your RNA. That one is the fastest one because basically you do all synthetic steps, and Moderna in 66 days was able to get to go into the clinic. Others took the synthetic gene and put the synthetic gene into a viral vector, mostly adenovirus. And there takes a little bit longer because then you have to grow the adenovirus, but it's just a little 1 month longer. So in 3 months, Oxford and AstraZeneca were able to be in the clinic. Others took the same synthetic gene and went through a more traditional approach. They engineered mammalian cells, insect cells, or plant cells to make a recombinant spike protein. Then the recombinant organism was grown, in fermenters, the protein was purified, mixed with an adjuvant, and then going to the clinic. So this one takes longer, it's more traditional, took 6 months. And then all these things were put in the clinic, and immediately in the as soon as they were in the clinic for phase 1 already, we knew how they were behaving. And we saw that the viral vectors were inducing antibodies, lower titers than RNA vaccines. And the RNA vaccines were inducing pretty high antibody titers, but some of the proteins plus adjuvants were inducing a little bit higher antibody titers. And the question was, will these antibody titers be able to protect? Our prediction already very early on was that all the vaccines were going to be protected. The reason is in this black arrow. This black arrow shows the range of neutralizing antibodies that were found in people that are convalescent from the disease. So the idea was that if the vaccines induce antibodies above this black line, the vaccines were going to be effective. Shortly after, we got the efficacy data, which showed that that was actually true. And even the viral vector vaccines were able to induce 60 to 90% protection from infection, while the ones with higher antibody titers got 95% efficacy. So all vaccines were able to protect from infection, but the ones inducing lower antibodies were a little bit less efficacious. On the other hand, all vaccines were able to give a very high protection, more than 95%, against severe disease and against mortality. So all vaccines were working well. And here we were at the end of 2020, and we are very happy because the vaccines were there, they were efficacious. Now we're going to get out of the pandemic. But at that time, the variants started to arrive. They started to arrive, first one, second one, then the Brazilian one, then the South African one, and then the Indian one. So what was going on with the variants? Well, what was going on is the following. Beginning of 2020, the virus was basically conquering the world, going into infecting all people. None of the people had antibodies to the virus. So the virus was just infected, didn't need to change, was happy, and was just spreading and conquering the world. Then mid-2020, the virus started to meet some people that had already been infected before. So they had some immunity, what we call natural immunity. Immunity induced by the infection. That immunity induced by infection was a kind of jail for the virus because the antibodies were basically neutralizing the virus that couldn't grow. But the immunity was pretty weak, and the virus found immediately windows out of this prison. And the windows were mutations in the spike protein that allowed to evade from the natural immunity. And the most famous one is the mutation E484K, but all these mutations allow the virus basically to come out with different variants and to start to circulate around the world again. And so the variants have been around the world, but then in the meantime, the world has started to be vaccinated. Now, the jail imposed by the vaccination is much stronger than the jail induced by the natural immunity. And so in the populations which have been vaccinated, which is Italy, but a lot of Western countries, basically, the virus stopped circulating. And however, so what was the prediction? Because now the jail of the vaccine immunity is much bigger, so the virus cannot grow. But unfortunately, after the 2 doses of vaccination, the immunity induced by the RNA vaccines or the viral vector vaccines, basically, the immunity obviously decreases. And we know that 5-6 months after the primary immunization, people can get infected again. Although they don't get severe disease, much lower hospitalization, much lower mortality, but they do get infected again because the antibodies go down. And the question is that normal? Is that what we expect? And you know, there are so many people debating about this and all saying everything in one direction or the other. But this is exactly what we'll expect if we go back to a textbook of immunology. In a textbook of immunology, basically, this is the red curve is the immune response in antibodies that you get after primary immunization or after secondary immunization or after a boost. So the first 2 doses are primary immunization. So what you get is an immune response, you get a peak, which is protective, but then the immunity wanes very quickly in 5-6 months. And that's where we are now in Italy. So what happens at this point? At this point, I mean, you can be infected again. But at this point, if you give a third dose, basically, the third dose is going to give a boost, and this boost is going to be induce antibodies which are much bigger, much higher than the previous one. And they're going to also decrease, but they're going to decrease to a level which is much higher than this one. And so protection is going to last much longer. That's what the textbooks say. So is that what happens? Well, here is what happened with the Moderna vaccine. After the primary doses, you get, these are the neutralizing antibodies against the wild-type virus, the Wuhan, and lower antibodies against the variants. A few months later, basically, the primary immunization has gone basically to zero compared to the variants, and even against the wild-type is very low. But then if you give a boost, you get much higher antibody titers, more than before, both against the wild type, against the variants. So the immunology textbook was right. In Israel, they started immunizing before any other country, and they got the reinfection in vaccinated people much earlier. And so basically, at some point, they were getting the infection in the vaccinated people. And at some point, they decided to revaccinate the people 60 years of age and older. And immediately, basically, the reinfection went down. In the people below 60 years, which will not get a third dose, infections continue to go up. So what was going on? What was going on, basically, is something that we published in a paper which is out in Nature as a preprint right now. And it's basically the primary immunization, where we've been looking at the B cells producing neutralizing antibodies. After primary immunization, these blue dots, basically you see B cells are not that many, and they're not very potent. And they are not very good against the variants. You see all these things, these B cells are not able to recognize the variants. After a booster or after a hybrid immunization, basically, where you get many more memory B cells, neutralizing antibodies, you produce neutralizing antibodies, much more potent, and they are many more against the variants. So basically, the third dose or the secondary immunization induces much better, much stronger neutralizing antibodies, and which cover not only the wild type but also the variants. So that's what's going on, and that's more or less the end of the vaccine story. Basically saying that if we give a third dose, will avoid reinfection of the vaccinated people. The virus will circulate less and will be able to control the virus. So the vaccines is a success story, an incredibly fast deployment of vaccines has been possible thanks to the RNA vaccines. And there is only one thing which really has not been working, and that's the fact that all the doses have been used mostly in rich countries. Low-income countries got nothing yet. And that's this pandemic has really increased the inequality in vaccination. And this is a big problem because the virus that circulates over there not only is basically a disaster for those populations, but it's also a disaster for us because new variants are going to be generated, the economy doesn't grow, people die, and unless we solve this problem, basically our world will never be in a good shape. That's the story of vaccination. Now let's move a little bit to passive immunization. Passive immunization is nothing new. Actually, the first Nobel Prize in medicine, Emil von Behring, got the Nobel Prize because discovered that the serum against diphtheria toxin was saving lives. This was 1890, so under 30 years ago. And passive immunization with antibodies from horses or purified from the plasma of people have been used a lot. Indeed, when the pandemic started, many people rushed to use plasma from convalescent people to try to cure people. Didn't work because, as you have seen, the natural immunity was not really strong. And so it did not work. Today, passive immunization means monoclonal antibodies. Monoclonal antibodies are nothing new. During the last 20 years, there have been more than 100 human monoclonal antibodies have been licensed and are available. But they're mostly available for autoimmune diseases, cancer, and they're not really available for infectious diseases. And the reason why they're not available for infectious diseases is that, again, technology. Back in 30 years ago, basically, when we started to make the first human monoclonal antibodies against HIV, the potency of these antibodies was so low that really were absolutely useless. Then 10 years later, we were able to do antibodies that were 10 times better, then 100 times better. Today, we can make human monoclonal antibodies, which are 1,000 times, sometimes more than 1,000 times better than the ones we used to make. So when the COVID came, we said, we're going to use this technology to make antibodies. And that's what we did here in Toscana Life Science in Siena, but many people in the world did the same thing. We did that in academic lab, but many companies like Regeneron, Eli Lilly, AstraZeneca, GSK with Vir, they also started to do that. And I'll tell you our story. So basically what we did, we took the blood from people that are convalescent from the disease, from the Spallanzani in Rome or from the hospital of Siena, and we basically sorted the B cells that will recognize the spike protein. And you see, we got more than 4,000 different B cells. Then we looked how many of those were actually able to neutralize the binding of the virus to the receptor, and they were much lower. So most of the B cells recognized the spike protein were not able to neutralize the binding of the virus. Then we asked how many actually are able to neutralize the infection of the virus. And we found out of 4,000, approximately 450 were able to neutralize the virus. And then we looked at their potency. Most of the antibodies were pretty low potency. They required 500 nanograms or more to neutralize the virus. Then we wanted really very potent antibodies, and we looked at those indicated by arrows that will be the ones that will neutralize the virus at 10 nanograms per ml or less. This is 10 nanograms, and this is less. So we picked these three. Eventually, we selected one, and we made a cell line that will produce it, and we characterized. We made sure that was not only neutralized the wild type Wuhan virus, was also was able to neutralize the alpha, beta, gamma, and delta viruses, which was able to do. And then obviously we asked whether whether in addition to be able to neutralize the virus in vitro, was also able to neutralize the virus in vivo. So we did in hamsters, basically, we gave the antibody and then the virus, and we saw that if we gave the virus along with without antibody, like in the red line, the animals lost weight, while if you gave the antibody before at different doses, the animals regained weight and they were fine. And more important, at 3 days, there was no virus in the lungs. Then we also do the therapeutic trial, basically, where we gave first the virus and then we gave the antibody. And this time, obviously, the animals lost weight, but then the animals that did not have the antibody in the red line took much longer to recover, while the ones that got the antibodies were much faster. And again, at 3 days, there was no virus in the lungs of the animals. So we had an antibody that was working in the animals in vitro, an antibody was working in animals. We did, in collaboration with people at the Scripps, the structure, the antibody in blue, and we saw that was recognizing the receptor binding domain in any position, up and down. We fully characterized with the crystal structure, and we realized that this, our antibody, which is this one, is the one that recognizes the receptor binding domain in a very small area. Most of the antibodies recognize an area which is 1,000, 1,500 square Armstrong. And so, and you see, these are antibodies, much bigger area, which is recognized. But if you recognize much bigger area, you are more susceptible to being, not be able to be affected basically by the mutation in the variants. While if you have a very small area like this, it's much more difficult for the variants to be able to overcome the neutralization. So that's what we did. And then we compared our antibody with all the others that have been developed. And we found that our antibody is the most potent, which has basically been developed and gone to the clinic, is able to neutralize all the variants at less than 10 nanograms per ml. All the others antibodies are basically are few, 3, 4 times, 10 times, sometimes 20, 50 times less potent than what we have. So then we put this in, we engineer this antibody to make sure that we'll inactivate the function of the FC to make sure that was not going to enhance the disease. I'll show you it's effective against the variants. And basically, since it was very potent, we had the possibility to use a very low dose, which could be given intramuscularly instead of all the other antibodies that were given by intravenous injection. Then we started a phase 1 clinical trial with 50 milligrams, 100, 400 given by injection. The things went well. And here you see that when you give the injection, basically the antibody, a few hours later, basically 4 hours, you already have very high titers of neutralizing antibodies. And those last up to 30 days or more. And if you want to see how the antibodies, neutralizing antibodies compared to vaccination, you look at these 3 things. These are neutralizing antibodies after injection of 50 milligrams, 100 milligrams, 400 milligrams. These are the antibodies levels which are induced by primary vaccination with the Pfizer vaccine. You see they're 10 times lower than what we get by injection with this. These are antibodies after the 3rd dose or after immunizing people that had previous infection. And you see, basically, with 100 milligrams and 400 milligrams, we get antibody levels which are much higher than that. So the antibody way is a very an immediate way to get very high antibody neutralizing antibodies. And with those data, basically, we started a phase 2 clinical trial, which is going on right now, and is a double-blind, placebo-controlled trial with 100 and 400 milligrams against the placebo. We are planning to enroll 800 people altogether, but 400 people will do an interim analysis. Right now, we are approximately 300. And we will basically, as a primary endpoint, we'll have the time to negative swab. So we expect our antibody will make people negative very quickly. And will avoid, if people become negative quickly, obviously they will not go to the hospital, they will not have a severe disease, they will not die. So that's what we expect. Now, all this has been done with the collaboration of the Italian government, Ministry of Health, but mostly with an academic lab. In the meantime, the big companies, mostly AstraZeneca, Regeneron, GSK, and Eli Lilly, they've done 3 trials for each of the antibodies, and they got the first data, and they've shown that the antibodies basically are able to prevent severe disease and mortality if given very early after the infection. And so we know the antibodies work, and they are available. These antibodies are available in most of the hospitals in Italy, and hopefully our antibody will also be available soon. With that, I'm getting to the end of my presentation, and I want to basically conclude with one slide. I think what I've shown you is that the science has done an incredible job by providing in a really an incredible fast track the vaccines. We've been able to basically prevent lockdowns and a lot of mortality in our countries. We have been able to reopen a lot of things to be free again. We have been able to restart the economy, and through the monoclonal antibodies, we've been able to prevent mortality in those that get infected. And obviously, monoclonal antibodies and vaccines are complementary, because vaccines are there to prevent infection. But once for those people that get infected, the vaccines cannot work. So you need therapy, and the antibodies can be a good therapy. And so basically, my conclusion is that thanks to the science, we are regaining our liberty. Why do I put down the liberty statute? Well, because this lockdown, this virus has taken away completely our liberty, the liberty to go out, to go to work, to walk, to play sport, to go watch games, theater, cinema, everything. Visit relatives. This virus has taken away everything from us, all our liberty. And now, thanks to the vaccines and thanks to monoclonal antibodies, thanks to the science, we are getting all this back. And I'm so proud that the science has been able to deliver this to our world. And with that, I would really like to thank you for your attention.